Good morning, ladies and gentlemen. And it's my great pleasure to introduce today's speaker, Professor George Church. As you know, Professor is an expert in the field of including sequencing and the genome database. He is currently a member of the National Academy of Science and Engineering. And in particular, he established so-called PGP Global, Personal Genome Project Global. I think this is a very important international genome database, in particular an open source database. So I think we are very fortunate and great to have Professor George to have a talk today, this morning. Professor George, please.
Thank you very much. Very nice to be here and to be talking about the Personal Genome Project, among other things. So the topic is reading and editing genomes and the technology, where it's been recently and where it's going. Everything that I mentioned having to do with human tests, any kind of experiments involving human cells, was through the Personal Genome Project. And that's because that's proper consenting mechanisms, and it is open access. So anything we do with it, we can share the cells freely without onerous MPAs. The data is freely available, like Wikipedia. Literally, you can use it without asking permission. It is also used by major government organizations like the NIST and the FDA, that you've probably heard of. Genome in a bottle is standards for genomes, and those involve trios, mother, father, and child. It's also used by the NIH ENCODE project and many other NIH projects, as well as assessment of interpretation tools. It's international. The various organizations are down at the bottom there. And that's where our human cells come from, and organoids we'll get to in a moment. Now, technologically, it's actually relatively easy to develop technologies these days, partly because there's these exponentials going on, factors of 10 on the y-axis here. Going back to the 1970s when I started, around 1984 to 88, we developed the concepts of multiplexing, barcoding, and reprobing, which were fundamental for what later was the next-gen sequencing. This was pre-genome project, but nevertheless set the stage for post-genome project, so-called next-gen. We've improved both reading and writing 10 million-fold thanks to multiplexing, and the slope changed from a doubling time of about 2 years to a doubling time in months. And we even have we brought the cost of genome sequencing down into the sub $500 range. But the price, there's at least one business model where the price to the consumer might be as little as low as $0 at Nebula Genomics by engaging the research community to pay for it. Now, multiplexing is a very simple concept. It's not parallelism where you make copies of instruments. It's where you shrink and combine reactions. So in a single droplet, one drop can handle somewhere between 100 and 1 trillion reactions for about the same effort as one reaction. So you can imagine the economy of scale there. And this is based on telecommunications dating back to the telegraph and dating forward to optical fibers. But this is molecular multiplexing, not optical multiplexing. Now that helps us lower the cost of reading and writing DNA, both research and clinical, but therapies, and in particular gene therapies, which are now the most expensive therapies in history, they need a boost, and they need a reduction in price as well. And these are two thoughts that I have on lowering the cost of therapies in general. Gene therapies are considered cost effective for orphan drugs for rare diseases. But here the two, I think, fairly obvious, once stated, is that instead of focusing on rare diseases, so you have a fixed cost, and in the rare diseases you get a small number of people who benefit, and you have to pay a large amount per person. Focus on common diseases, and many rare diseases are heavily impacted by common diseases, which I would say the most common disease is aging. So many diseases, both common and rare, are affected by aging. And so if you can get a single drug or even a single combination therapy that reverses aging, then you can greatly reduce the cost of that therapy in many diseases. But then that still leaves the rare diseases that are not fixed by aging reversal. And for these, I would recommend that prevention is better than cures, more cost effective anyway, and in some cases less invasive. And the ultimate prevention with the least amount of negative information, bad news, is early dating or matchmaking. And that's proven successful. Now, give some examples of multiplexing, this pooling. We can do multiplex synthesis and testing of things like AAV. The most well-characterized gene therapy and safest and most commonly adopted gene therapy vehicle is AAV. And we do rounds of multiplex synthesis where we're not making random viruses, mutagenizing them, we're making designed viruses. The computer is learning what it knows from previous experiments, previous data sets, to design new viruses. We've made over 1.2 million different designs and synthesized them, and you can inject them in a pool, so they're all together. We don't do a million animals, we do one animal with a million viruses, and then we take samples, biopsy specimens from every tissue that we're interested in getting delivery. We want to study delivery, and then we analyze them. And this is work from Pierce and Eric, who have formed this company, Dino Therapeutics, to provide these gene therapies to many different companies and different therapies. So this is the result, sort of results that you get from different tissues. This is not, this is by no means a complete data set. This is just a tiny fraction of the genome here, starting at position 570 along the x-axis, and along the y-axis are all the different amino acid codon substitutions. At every position in the entire virus, we've substituted every possible change in the genome. And you can see this heat map ranging from red to blue, ranging from better than wild type to worse than wild type. And you can see these ratios change with the position in the virus, the type of amino acid, and the tissue. These are 6 of the tissues, but we have more. And we've done this for mouse, we're doing it in primates, and then we're trying to do it in humans. Now, as an example of what you can get from this very comprehensive analysis, so we can make a large number of substitutions at once. So we can go, we can take a virus that, if we do random mutagenesis, it's hard to get more than 4 substitutions without killing the virus, killing its functionality. Or we can do up to 25 or 30 substitutions, insertions, or we can do them systematically throughout the virus. And in the systematic process, we found a new gene. And finding a new gene in a virus that's studied this much, which is so critical to therapies, and it's so small, is an unusual thing. And this is the position of the new gene. When we not only have shown that it is present in the original strain, but its sequence is conserved over many different AAV type serotypes. And when we look to see where it is expressed on the plasma membrane of every single serotype. So it's not only is its sequence conserved, but its functional context is conserved. It is essential under a variety of viral growth conditions, in particular competitive growth conditions. Now that's gene therapy. I want to now talk about cell therapy. These are somewhat related. And in particular, I want to talk about the universal cell therapies, or allogeneic, where you can take cells from one person and give them to another without matching them. The matching has been required ever since the dawn of surgical transplantations since 1954, when it was first done. But as of 2013, there now is an alternative where no matter what you're transplanting, this is CAR T cells, but it's also true for a variety of other cell therapies. You might have to make not one edit to one gene to make the cells acceptable universally or allogeneically, but you might, here's 5 different mutations from the literature that different groups have used in various combinations to make your incoming donor cells compatible with the recipient. And CAR T cells are used to fight cancers, but there are other cell therapies. And the point of this is that you might have to do multiple genes, and you don't want to feel limited in the number of genes. You don't want to be limited by the technology. You want the technology to be efficient, on target, and very little off target. Because the more genes you add, the more off targets you're going to have. So we studied this at the extreme, of course, at the extreme. And that is not just the number of edits you would need to make a human, any human, compatible with any other human, to make an animal compatible with any human. And here, it's more than 5. It's about 43 genetic changes that's required to make pigs like this that are capable of transplanting almost any organ that you would normally transplant between humans. And so we need, the categories are sugars, so there's at least 3 sugars that are different between pig cells and human. There's clotting factors that are incompatible with the tissues. The major histocompatibility is the classic, class 1 and class 2. Other immune functions that help adapt for transplant. Complement is another cascade in the blood. And finally, or initially actually, the endogenous retroviruses. This is something that, despite a $1 billion commitment in the industry 20 years ago, this is an old idea, and the FDA initial enthusiasm, they became very nervous that every cell of every pig in the world releases retrovirus, and retroviruses are not typically benign. They will infect human cells, and then they will do secondary infections of other human cells. So these are really not things you want to have in immune-compromised patients in particular. You do not want them evolving into a zoonotic disease like Ebola or swine flu. So we undertook this in 2015 in transformed cell line, just a cell line, not a pig, but pig cells. And that was very easy, surprisingly easy. Then we had to redo it with normal cells from a different strain, smaller number. So the first one was 62 retroviral genes, and then for the 2017 paper, it was 25. But it was harder, actually. 25 was harder than 62 because there were normal cells, and normal cells are much more sensitive to editing. Editing, the kind that we're doing, I call genome vandalism, because it's not precise, but it knocks out the genes. And then this is advanced enough so we have now a herd of pigs in both China and in America with these 2 companies, Kehan and Eugenesis. And I'm sure that was the work of Luhan Yang and her team. Luhan was one of the co-inventors of CRISPR, and then she went on to invent a lot of this technology having to do with pig transplants. Now this is why I say it was harder the second time. The first time we got quite a number of single-cell clones that had multiple edits, 62 edits. We got none that had even 25 edits in the normal pig cells, normal pig fibroblasts. But if we include basic fibroblast growth factor and P53 inhibitor, then we get almost everything is highly edited, up to 25 edits. And we take the best clones here that are what you want on target, very little off target, and then do nuclear transfer, somatic cell nuclear transfer to establish new pigs. And we use that to fight senescence as well. So as we're making these 43 engineered genes, the fibroblasts start to senesce, as all fibroblasts do, and just by running it through the embryo again, we can rejuvenate them. And that works quite well, and that's why we have these pigs. And I should mention, these pigs are in preclinical primate trials at Massachusetts General Hospital. Jim Markman is running the team, and they're looking very promising. 9-month survival. We're testing kidney, liver, islet cells, and so on. So now, the reason that that was possible was CRISPR. CRISPR is not the only tool that we use, but it is one of the main ones we use for engineering those pig cells. And multiplex editing is still quite challenging with CRISPR. So some of them we put in as large cassettes where we can make multiple changes at once with a big cassette. And other ones we're doing genome vandalism. And what I've already emphasized how much harder it is to do normal cells, and I just point out that in the original 3 papers in 2013 that introduced CRISPR, the technology for human cells, most of them since, if you read carefully, do not use normal cells. So when you're reading the CRISPR literature, look very carefully that they're using normal human cells. And the ones we use were from the Personal Genome Project, induced pluripotent stem cells, and we continue to use those for all the other experiments I'll talk about today. Also, avoiding off-target was not talked about in most of the early experiments, but we did it in our experiment. And then the idea of using libraries is multiplexing, and we use that extensively. But anyway, Feng Zhang and Jennifer Doudna and I, along with David Liu and Keith Young, took these early results in beginning of 2013, and by the end of 2013 had founded Editas, and by this summer, this year, we have the first in vivo editing therapy for LCA10 in the retina. So that's pretty fast from an idea to the clinical trials. Now, we were emboldened by our ability to engineer normal pig cells at repeats, these endogenous retroviruses. So this is a repeat table for humans, but humans have a few more endogenous retroviruses. But we looked at these are the repeats that have historically and even today mess up the sequencing, the reading of human genome, reading of animal genomes in general, and they're even harder to do writing or editing or genetic analysis, because if you have a multi-gene family where you have multiple copies of a protein or some other repetitive element, and you want to show that that is has a function when you knock it out, you really have to knock out all the copies of it. And so making point mutations in all the copies of each of these repeats is challenging. But we were, since we did it for endogenous retroviruses at 25 to 62, we decided to tackle lines, these long interspersed elements. And there are about 26,000 of these that we tackled per genome, per haploid genome. And these are not just a nuisance or an annoyance in the genome that are hard to sequence and hard to mutagenize. They're also involved in senescence, neurogenesis, cancer, and inflammation. And these are articles not from my lab, from other labs, and there are more. We could list more. It's hard to do these experiments. It's especially hard to establish cause and effect genetically if you can't edit all of the elements. So that's what we're attempting to do. And we started with the line elements, line 1 elements in human, and we found very quickly that CRISPR and other CRISPR nucleases were very toxic. They're very, if you have more than 100 repeats, it's incredibly toxic. Even, not just double-strand breaks, but even a nick is toxic. Even the nicks that occur, so we switched to deamination, and even the nicks that occur when you deaminate a C to a U to get the T, deoxy-U is repaired, and almost any repair process in the cell causes a nick, and that is apparently toxic. So the recipe for making this level of multiplex editing, where you have hundreds or more of edits, you need to have all of these right. I already mentioned 4 and 5 in the pig experiments, but we also had to eliminate all the nicks from so-called dead Cas9 or defective Cas9. It's defective with respect to nicking, but it is still potent in terms of carrying the deaminase domain for either cytidine or adenosine deamination. Now, A deamination is not subject to repair, and so it actually has better, lower toxicity. It has another advantage over C deamination. C deamination, the apobec that's typically used for the deaminase will bind to the genome itself, and so it raises the off-target by a factor of 20. I mean, off-target hasn't been a proven clinical problem, but it certainly is disturbing that it increases that off-target. So the A is what we got the best results with, and by we, I mean Corey, Oscar, Khalid, and Verena. This team took on what looked like it might be a very challenging task and made it work quite well. This is an example of the number of edits we got with these various different constructs. So the first three are cytidine deaminases, and the next three are adenine deaminases. And the one that worked by far the best was this non-nicking adenine base editor. I should mention that the deaminase strategy, the targeted deaminase, started again with Luhan Yang, the same Luhan Yang I mentioned earlier, and then was greatly accelerated and improved by the David Vu's lab, both for C and A. The A was particularly clever use of adenine deaminase from transfer RNA metabolism. In any case, if you look at the outliers here, since almost all the work we do is clonal, meaning we take the best clone, the best single cell out of derived clone, we got 13,000 edits per genome, which if for a diploid genome would be about 27,000 edits, which is our current best effort, but we're moving on to a even more prominent family. Well, first we're trying to make sure we get every one of these line elements. We're also going for alus and centromeres, which have about over 1 million copies each in the genome. We haven't done that yet, we're just starting on it. So that's where we stand on the highest level of multiplex editing, and we think we're going to see that see this more and more often in research and clinical use, where I already showed you some examples. It's not advancing. It was advancing. Oh, this here, okay, that's good. That's good. Okay. So, another example of hyperediting. So the examples I gave were having universal donors for transplants. Another example is making virus resistance. We want our transplants and various agricultural, plant and animal, we want them to be multivirus resistant. Even manufacturing, you can have many billions of dollars lost when your company gets infected and the manufacturing process has happened at enzyme. And the one way we found to make, we think, we're capable of making any cell or organism resistant to all viruses simultaneously on paper, on theory, is to change the genetic code. The viruses don't share that much in common, but they do share that dependence on genetic code. We've shown this to be true for one industrial organism, E. coli, and almost all viruses, but certainly multiviruses resistance, by changing 321 codons genome-wide, which allows us to delete the otherwise essential gene that does release factor at UAG stop codons. So the stop codon UAGs are changed to a synonymous UAA codon, and we can do that genome-wide, and it's rather efficient for non-standard amino acid incorporation for biocontainment, and notably multivirus resistant, about 92% of viruses. We think we hypothesized that we could delete any pair of codons that the transfer RNA wobble hits pairs of codons. If we could get rid of any tRNA-mediated pair of codons, that would be enough to make it resistant to all viruses in a way that they cannot revert, they cannot escape. We're in the process of testing that, and I'll show you where we're going with that. In order to de-risk you actually also learn something about the coding regions by looking at what codon synonyms, so two codons that code for the same amino acid but with different nucleic acids. We swapped every codon that could be swapped. So there's 64 triplet codons. 2 of them are unique, methionine and tryptophan, and 6 of them, sorry, 3 amino acids, leucine, serine, and arginine, have 6 codons. So 2 have 1 codon and 3 have 6. And we changed every possible codon, all 62 of them, for a large number of essential genes, essentials being the hardest ones to do. And all this blue is where we were completely successful, and in gray were places that still needed to be sorted out. But this allowed us to reduce the risk of changing 7 codons, the original one in red, and 3 more. Remember I said leucine, serine, and arginine have 6 codons, so there's a lot of place to swap. This leucine goes down here, this serine goes up here, this arginine goes up here, so they can be synonyms. And then another group, Jason Chin's group, picked another serine codon, which can be mapped, remapped. None of us have yet finished it in the sense of deleting the tRNAs so that we can free up any of these pairs of codons. But hopefully that'll happen soon. Millie Ostrov is championing running this project. So I've given a couple of examples of pathogen resistance and immunity preservation. We have examples of senescence resistance, cancer resistance, cryopreservation, and DNA damage resistance in various organisms. And we use, these are, inspire us to try to make organs that are enhanced. We don't necessarily want to make, superhumans. We just want to make organs that will do their job by not succumbing to pathogens, senescence, and so forth. And for cryopreservation, here are 2 of our most amazing organisms that inspire us, this midge and the tardigrade that can survive liquid helium temperatures and severe desiccation. So these might be very convenient for storage of organs. Each of these things, as I said, have been demonstrated in animals, and we're just transferring them into pigs to make better organs. That's happening in my lab, not in the company's Key Hunter and Genesis. I mentioned the rejuvenation that we do routinely while we're improving the pig genome. We also want to be able to do it in the form of gene therapies for aging reversal. We focus on aging reversal rather than longevity because it's hard to convince the FDA that you have longevity without doing a very long clinical trial. So instead, we do gene reversal on well-characterized, well-known diseases. So if you get multiple diseases that only affect older animals or people, then you're probably getting at the core of aging rather than if you just get them one at a time, you might be just dealing with the idiosyncratic symptoms. And so the 5 that we've chosen so far, and this list keeps growing, is some of these are kind of accelerated aging for convenience. A high-fat diet, obesity, where they've doubled the normal size, type 2 diabetes, osteoarthritis, cardiac damage recovery, and kidney disease. And we started with 300 genes and whittled down to 45 that were non-cell autonomous, and then found 2 or 3 genes that in combination would hit all 5 of these diseases. And we're now, 2 papers are coming out in PNAS in November, and we're extending this now to dog clinical trials, and a company, Rejuvenate Bio, is taking charge of that. Noah Davison was a postdoc in my lab, as was Pedro de Magalis, who developed the database. Noah did these gene therapy trials. Now, another thing we can do that's relevant to senescence is in human cells, we can reprogram them both epigenetically and genetically. So we can reprogram them epigenetically at very accelerated rates. We can go from stem cells to almost any cell type you want by studying the transcriptome and using transcription factors, as you'll see in a subsequent slide. We can go within 4 days with 98% efficiency to a variety of different cell types. And we can do it to complex organoids, which are multiple cell types, and we can recapitulate at least some of the abnormalities of either transcription or cellular abnormalities in as little as 2 to 4 weeks. For diseases that have very late onsets, take 70 years to develop in a human being, we can see abnormalities in as little as 2 weeks. And you'll see in the next slide how dramatic this is. This is thanks to a beautiful collaboration with Bruce Yanker's lab, who's in our department. And we have this first paper in Cell Reports on late onset Alzheimer's, we've also done unpublished schizophrenia and bipolar and have quite significant results in all cases. But I'll show you Alzheimer's, since this is something you can easily check by going to the literature. Here we have transcriptomes that have been clustered in the computer, both by case and control. These are age-matched controls on the left, cases on the right. You don't need me to tell you that they're dramatically different in a variety of different genes in this RNA clustering. But the cases and the controls are age-matched, but they're genetically quite different. They have 1 million differences or 3 million differences between any two random individuals. So we wanted to be able to do both the epigenetic reprogramming, where we can get brain-like organoids, and epigenetic reprogramming for old brain organoids. But also, we'd like to be able to do genetic programming so we get isogenic lines, meaning they are identical at 6 billion base pairs, but differ at 1 base pair. And the one we tried in this, we've done quite a number of these, but just as an example, we've done E4. ApoE4 is a risk factor for Alzheimer's, and E3 is a normal allele. And so you can make isogenic lines, and again, you do the transcriptional profiling at 2 weeks, and you get this dramatic difference between now it's not case and control, but it's isogenic cell types. Now, the reason all of that was possible was because that we can do this epigenetic change. It's not that it's genetics with CRISPR, but epigenetics with transcription factors. And we used transcription factors in an expression library, and these didn't exist to our surprise when we started this, so we sort of tediously over the course of the last 3 years have accumulated a complete library now, which is available to anybody from Gen 9 as soon as the paper is published. And I noticed that one of you has an AdGene t-shirt on, nice genes. That's what AdGene's about. It's a nonprofit that distributes these, and we have expression of every single human transcription factor, including splice, a subset of the splice forms in AAV, lentivirus, or piggyback. We like the piggyback the best because it has the largest packaging size in the hundreds of kilobases, and it has the highest copy numbers, the highest expression level. And so it's enough to like blast the cell out of its epigenetic stem cell state into a new state without any additional factors, just transcription factors. And we do this, we can either screen the whole library and see what they can do individually, or we can make combinations up to 20 at once by doing transcriptome targeting, where we analyze the transcriptome, the RNA of the target cell, and then make those, only those in various combinations, those transcription factors. And this is work of Alex and Parastu and coworkers, and this paper is in review, and they started a company called GC Therapeutics. And you can use these in various combinations, so you can do them singly. You can get pure tissues, 98% pure, but you can also do combinations. For one of our favorites is combination of oligodendrocytes, which wrap the axons of neurons. So oligodendrocytes, neurons, and endothelial cells, we'll see in a moment. They wrap it the number of times you would expect a normal. They don't, not fewer or less, is quantitatively correct and geometrically correct. And then this can be used to study demyelinating diseases, both in vitro and in vivo. We can implant these complex organoids into mice, shiver mice that have demyelinating diseases, and then we can see impact on their functionality. One of the things that was limiting the organoid field, we did not start this field. There were pioneers like Gail Martin and Madeleine Lancaster. But one of the things that was lacking was vasculature. So with Jennifer Lewis and Mark Schuyler Scott, we developed a protocol for getting endothelial cells to mix properly, and not just mix, but spontaneously form capillaries, blood vessels that were about 5 microns inner diameter, just the right size for blood cells, and then showed that in the mouth, when these are transplanted in mice, they fill with mouse blood cells, as you would expect. just want to end on ways of analyzing these organoids and developmental biology in general, ecology. So organoids, normal development and abnormal, and be able to study the DNA, RNA, and protein and how it plays out in time, not just a snapshot, although most of what we do is a snapshot. We want to be able to figure out how to connect time with a snapshot. And some of you probably know about light recorders. They're not actually black, they're orange in your plane. And we've been championing for years that you can store data in DNA, and we have a nice startup company called Kern on this topic. But in this case, you can store up to 455, oh, that's incorrect, that's exabytes per gram. What's 1,000 among friends? But this is a different way of recording. In fact, we weren't really intending to record DNA when we started this, and by we, I mean Prashant Mali, who was co-inventor of CRISPR along with Luhan and Feng Zhang and so forth, and Reza Kalhar. And we used CRISPR in an unusual way, rather than a textbook way of making a guide RNA, which binds to the nuclease, cuts the genome, does genome vandalism, game over, doesn't do anything more other than target mischief. Instead, we inserted the PAM site, which is the part that's not in the guide RNA, into the guide RNA. So now the guide RNA becomes its own target. This is a very strange, it's like painting a bullseye on yourself and then shooting at yourself. But then after the genome vandalism occurs, game is over only temporarily because a new guide RNA is made, new nuclease, and it cuts itself, and it keeps cutting itself, generating information. This genome vandalism is fairly random. Usually that's a bug we're using as a feature, we're generating information. And these guide RNAs, of course, are all part of the transcriptome at this point, and so we can detect them in situ. So these are almost everything, if we possibly can, we try to do it in situ. So we retain the full three-dimensional information, multicellular and subcellular context that a pathologist cares so much about. So, and that information is on 40 to 60 different sites of the genome. The mouse are crossed with a Cas9 mouse. You don't want the Cas9 around all the time. They don't actually look striped like this. This is a cartoon. But you can tell things about development because you've got now a lineage. Every cell in the body has recorded all of the developmental information from single cell egg to billions of cells in the adult. And you can tell things like, one of the things that we discovered is that the left-right decision in your brain is later in development. Not necessarily your brain, mouse brain, is later in development, probably yours as well, than the front-to-back decision. And we calculate that we're storing on the order of a terabyte of information, 4 trillion bits, in just a billionth of the mass of the mouse. That would be very hard to do that with electronics. It's a 30 nanogram storage device of a terabyte. Now we read this out. I mentioned in situ. I showed a picture of in situ already. This is really in situ on steroids. We can do DNA, RNA, and proteins in pathological situations, normal organoids, and so forth. And there's 2 instruments off the shelf. You don't have to build it yourself. You just these are basically the cost of a regular microscope, but there's so much more than a regular microscope. Every pixel, every voxel in principle can have a barcode that tells you which DNA, RNA, or protein exists at that pixel or voxel with pretty thick specimens. This is not spatial transcriptomics, which is low resolution and not very thick. This has resolution up to 12 nanometers. This is incredibly high resolution. Here's an example of a single cell. Each dot is an RNA, 4 colors, ACGT, repeating through the cycles of next-gen fluorescent sequencing. Then the computer can recite, restore, tell you what barcode each RNA was at each site in these are PGP1 fibroblasts. So again, every human experiment was done with Personal Genome Project cells. Jay Lee and Evan Doherty were postdoc and graduate student, and Jay's at Colesby Harbor, and Evan has helped start ReadCore, one of those 2 companies. So just last two examples, that was RNA. I want to show you DNA and protein, so you can do all three simple dogma macromolecules. This is work from Ting Wu's lab. I collaborate with her a little bit, but this experiment was entirely in her lab. These are the postdocs that did the work, and these are some of her PI collaborators. Hung Yen in particular I want to call out because he's worked with us as well on super resolution proteins. We'll see that in a moment. To validate this, we use normal human pedigree, sort of classical Mendelian mapping to get haplotype information. And this was aided by, again, Personal Genome Project, volunteer number 1 and number 595, which are de-identified as myself and my half-sister, Jennifer, who my mother was clever enough to make sure we had different fathers so that the haplotyping worked out well. So here, these are my cells, P2P cells, which have two chromosome 19s. So this is a single nucleus, two chromosome 19s, starting on a walk. You can walk through all of the chromosomes from telomere to telomere, so there's no loss of continuity. You don't have to worry about short reads. Here's 3 chromosomes read partially through. The Wu lab is working towards getting all of the base pairs of all the chromosomes. So this could be a new way of doing sequencing that has no gaps. Now, finally, I said RNA, DNA, now proteins. For proteins, we have to kind of turn it into a nucleic acid problem by changing it from a protein problem to antibody to nucleic acid by covalently attaching a 9-mer, 9 long oligonucleotide to the antibody, either these large antibodies that are recognizable as Y-shaped IgG, or a tiny domain, this so-called nanobodies, which is just one of these domains enough to recognize your protein. And then this little 9-mer allows us to do DNA exchange imaging, which is Hung Yen's lab developed this, and I'll show you that in just the next slide. Here's the super resolution you get from DNA exchange imaging, this new way of doing super resolution for these DNA labeled antibodies. Diffraction limited normal microscopy was totally inadequate for this task. And the task here is determining the pre and postsynaptic proteins so you can tell which direction your connectome is going. When a 2 neuron synapse, you want to know which direction the information is going. And we have up to a dozen proteins at once, up to 4 per synapse that are clearly labeled. And we can also tell whether they're excitatory or inhibitory. This is work with Yu Wang, who was a joint graduate student with Hung Yen's lab and mine. He's now a postdoc working on following up on this sort of thing. So that's proteins, and I will finish a tiny bit early and open it up for questions. I just want to remind or say thank you again to the Wu lab for the DNA work, to Jennifer Lewis's lab for the vascularization of organoids, and Jim Markman for doing the clinical transplants at Massachusetts General Hospital. Thank you very much. Plenty of time for questions.
Very thanks, professors, for the excellent and inspiring lectures. Are there questions from the audience? Yes, please.
There's a microphone.
Microphone.
Right.
Okay, so what happens now that we've optimized sequencing and eventually we'll optimize storage and editing DNA? What's left to do? What's on the horizon for the field?
There's so much left to do. I mean, first of all, we have to get 7.7 billion people sequenced for their inherited genome. We've done maybe 1 million and not very well. So no, we've never finished a genome sequence. I think in situ will increasingly be the method for looking at pathology, biopsies, and so forth. But we also have to do environmental monitoring. And for that, I think we need wearable sequencing devices. So like this phone has 6 cameras on it. Should have 6 cameras and 2 sequencers, I think, that are monitoring the allergens, the pathogens in the air, water, food, 24/7, should be doing all the time and reporting not just to you and your physician, but to the entire community, so we can do epidemiology at an unprecedented real-time rate. And I'm just getting warmed up in terms of what's left to be done. There's a lot, don't worry about running out of things to do, yeah. Other questions? Yeah. Middle there.
Hi, you speak of making plants less resistant to all viruses. Do you see doing that in humans, resistant to all viruses as well?
Well, it's a little harder to do in humans, but what we'll see first is organ transplants into humans. I think we want the organs to. If you lost your liver to hepatitis, you don't want the organ to get a trivial viral infection. And as we get more confidence and that works out well, and there's no toxicity, no negative consequences, then we will think of ways to get it into more and more cells in humans at risk, and then more and more cells in humans that as a preventative strategy. But that will all those steps will take time. Each one taking 10-year clinical trial. Yeah, back there.
Because drug resistance changes very fast, how could you handle this in gene therapy? And could we have any chance to create gene and edit into genome artificially?
Right. So let me repeat your question to make sure I've understood it. You can just raise your hand if I got it right. So you're worried that in drug resistance develops very quickly, and in what way can gene therapy help us deal with drug resistance? And I would include both microbial and cancer drug resistance. Is that fair? That's good? Okay. So, as you can see in this talk, I often wear different hats. At the beginning, I said, even though we're championing gene therapy, especially for the common diseases that are exacerbated by aging, where one therapy can help thousands of diseases, for many other diseases, prevention is far better than cure or aging reversal. And the prevention for Mendelian diseases is dating, and the prevention for cancer and infectious diseases is either, many cancers can be avoided by genetic counseling, and not all, by any means, and many infectious diseases can also be prevented by proper hygiene and so forth. I mean, this is a lame answer. Certainly I gave a powerful therapeutic that could avoid viruses, and viruses do become drug resistant. There are many of these diseases, these infectious diseases, and even cancers are organism specific. So if you study why certain organisms escape, then you could in principle do gene therapy toward the organism which is resistant to the various pathogens or the various cancers. Cancer resistance has been demonstrated in mice by transgenic models where you can greatly reduce, either by duplicating tumor suppressor genes or by anticipating the oncogene changes. One of the nice things about cancer, one of the better things about cancer than infectious diseases is cancer, the war begins fresh with each baby, and you get to see it over and over again. You get a chance to know where it's going. While infectious diseases keeps changing, your enemy keeps evolving, while the cancer is always the same enemy, and we get better and better at it.
In the first couple of slides, you mentioned about the tropism of the AAV, right? So if I catch the point correctly, it can determine the site specificity of the AAV delivery. But in some part of the body, for example, the eyeball, you can inject the virus directly into the eyeball. So the site specificity is very, very good. But to rejuvenate some organ or tissue, you have to deliver the virus systemically. So the tropism will determine the site specificity, right? But if you want to rejuvenate a large organ, so how much virus should you deliver? And is there any possible side effect for systemic delivery of the AAV virus? And how could we minimize the side effect and at the same time to increase the site specificity of the virus delivery?
Right. So aging reversal is the classic example where you would like to get almost every cell in the body, in principle, because every cell is senescing. Sometimes it's sufficient just to get to stem cells, and then eventually those stem cells will populate the entire body, so you can get a small number. There are ways. You had multiple questions. I'll just answer them as they come. The ways of reducing the toxicity. Well, first of all, AAV is by far the least toxic of the delivery mechanisms right now. That's why it's present in almost all of the approved gene therapy approved by the FDA. Nevertheless, a story that I didn't include because I included too many stories already. We have evidence that, or a method by which you can reduce the tiny residual of inflammatory innate immunity to the AAV particles with a short sequence, about 100 nucleotides long. It is a repeat of the human telomere, which causes innate immunity to drop by about 10-fold. And we've shown this in pig ophthalmologic surgery, in gene therapy and so forth. So you can lower the AAV toxicity, you can lower the AAV dose by targeting stem cells. Engineering the tropism helps you engineer that stem cell. And what we did practically, finally, the last part of the answer to your question, what we did practically for the first set that is appearing in the PNAS in November is we targeted the genes that were non-cell autonomous, meaning that their effect could spread throughout the body by systemic delivery of the proteins that are secreted from the cells. And that way you don't have to have efficient delivery to every cell for those particular ones. But in the future, we do want to get a sufficient delivery, at least to the stem cells. So that combination of inefficient delivery to a small number of cells and then a systemic production of proteins. A lot of aging is the slow, what looks like planned obsolescence, slow decline of key proteins that are needed to keep the entire body thinking that it's young. So if you can overproduce those proteins, that's what seemed to work in the mice.
I have one question. Could you tell us more about the personal genome project?
Yes, great. Thank you very much for asking. I sort of mentioned it all the way through. All of our human experiments are done with that. It is unusual in that there are many projects that are larger that are aiming to collect data, but it's very hard to share the data. And it's actually that some of the rationale is given is that patients don't want to share the data. That is if you talk to patient advocacy groups, that is rarely what they say. It is usually what the researchers say to protect their data. They want to hoard the data and profit from the data, but the patients want to share the data. And at least we want to enable the patients who do want to share. It's not necessarily all of them, but the more severe the disease that's impacting their family, the more interested they are in sharing. So this is a way that you can share it worldwide. There are no international barriers, there are no commercial barriers. So many of the other projects in the world that use human cells, human subjects, have a limitation for sharing for commercial use. And I think that is impeding progress of creating cures. So anyway, those are some of the features of the Personal Genome Project. Ethically, I think it's better as well, because when we started on this in 2005, there was this presumption that you could not let patients know that they were at risk for something you discovered. You could not give patients feedback on their data, even if you could save their life, because you might be wrong. Well, you're definitely wrong if you don't share the data with them. And you just need to, it's like all medicine, you need to figure out how to practically achieve the goals, which typically means, counseling them in advance and checking any result to avoid incidental findings or false positives. In any case, that was one problem. The other problem is you would promise them to keep their data secret while you're sharing it with hundreds of researchers, whether it's, even if you're not sharing it with the whole world, just the small number of researchers you're sharing it with is a risk. Somebody in there could leave it on a subway car or incorrectly let the data get out. In the era of WikiLeaks and ubiquitous information, it is a false promise to say that you can keep people's data secret. A better way of consenting people is to tell them, frankly, that it is possible, very likely that their data could get out, and they should not sign up for it unless they're comfortable with that. So those were two of the things that were innovative in 2005. Thankfully, they're less innovative in 2019.
Okay. Thank you very much for the inspirational talk. And I have a question about the prevention of rare diseases that you mentioned. Because what I know about rare diseases, like sometimes they're like early onset, or they're, when you found out this disease, it's already like too late, or even like when people are not diagnosed that they have these kind of rare disease. How do you make sure they would want to use like this kind of prevention approach?
Well, you nicely phrased it, exactly the motivation for doing it as early as possible. Many children are born already, the damage has been done. They're born with, say, microcephaly. Their brain is incredibly tiny. I mean, that is the challenge, is getting people interested in it. So, one of the things that I've been trying many different models for educating people, for bringing the price down so it's attractive, the $0 genome, maybe even pay them for their genome. Whatever it takes to get people to tweet about it, ask, why am I not getting my genome sequenced? The most recent one is I launched a dating app for the phone. It's not quite ready for your phone, but it's a company that's launched to develop a dating app. And it sounds funny, but it's actually very serious medicine. It's simultaneously not medicine and serious medicine. It's about $1 trillion a year worldwide, I estimate, that's caused by serious Mendelian diseases. This is not fun and games like, whether you have the right odor or the right personality. This is about serious Mendelian diseases, $1 trillion. And it's not serious in the sense that we're not actually giving medical advice. We're just saying, here's a list of people you're compatible with. We're not saying whether you're a carrier or not. We're not saying who you're incompatible with, just saying who you're compatible with. So it doesn't require FDA approval. And it's not like we're trying to escape FDA approval. It's serious. It literally shouldn't have FDA involvement. But it's very serious in the sense that it could, it's been shown for Tay-Sachs and a variety of Ashkenazi diseases that you can actually eliminate diseases in all the practitioners in a population. We just need to figure out a way to generalize that from 10 million Ashkenazi to 7 billion everybody else. And we're hoping this dating app will do it. Dating apps are becoming very popular, and this would just be something you don't even have to know much genetics or medicine to use this.
Thank you.
Is the PGP also including the extranuclear DNA sequence? And the other question is, when it goes from the United States to the global PGP, what is the government's attitudes or concerns you have experienced?
Right. Yes, it does include extranuclear. In fact, PGP is really whatever you want it to be. It's like Wikipedia. Say, does Wikipedia include articles about Serbo-Croatia? Okay, it does, if you wanted to. So we have, at one point or another, all the cutting edge methodologies, even things that are not not in current clinical practice can be included. DNA, RNA, protein, mitochondrial, you name it, in situ, things that are very, all the cutting edge things I described. And these, in principle, some of them are applicable to every person in the PGP, some of them are just a few, and if they look like they're working well and somebody wants to pay for it, we'll extend it to the entire cohort. Well, how do governments react? Well, some governments have rules that they don't want individuals to know their own genome, so that's a problem. Some governments don't want material to be shipped outside of the country. That's less of a problem. Some don't want data to be shipped out of the country. That's, again, a problem. To some extent, if people come to the United States or Taiwan, where we have democracy, then they can do whatever they want with their data, right? There's nothing to really stop me from taking my own blood, sequencing my own DNA, and posting it on Facebook, right? So, so that's not a problem. It's just in governments that are a little more anxious, and we're sympathetic. We're not trying to cause that kind of revolution. We're not trying to cause technological and medical revolution, but we think that it's best for everyone, and we're hoping that the governments will see, all governments will see the value of letting people know their genome.
I think, again, we thank Professor Church for such an excellent talk and Q&A. So I think I will close the section of this talk. Thank you very much. We
我们来照个相，哎，对对。
We will have a photo, so please sit.
好，我们就维持在原座啊，我们从前面往后照哈，对。那后面的一些老师，我们把第一排。